as I was trying to say, but I was muted, we annotate all this data, we integrate it, we build knowledge on where and when a gene is expressed from it, and now what can you do with this knowledge? How can you leverage it to learn more biology? So the first thing we have is that Frédéric just showed you the raw data pages. We have similar pages where you can leverage our expression codes. So of course you have the gene page that you've already seen, where you see for one gene the expression by anatomy, development, sex or strain, integrated, present absent cause, both from direct observation. So if someone took RNA from the cerebellum, you have the cerebellum, but then you also have the brain because it's propagated, brain being part of the cerebellum. You have orthologs and paralogs. I clicked earlier and showed you the expression comparison it leads to. And you have cross references to specific, notably UniProt and Ensemble when available. And you can also browse the present and absent expression codes in a similar way to what you can do with the raw data. So you can browse like this and see for all the genes filtering over species, experiments, and so on, I mean conditions and so on. So for example, here I have all the expression for the gene OXD8 in different conditions, and then you can get to the source data. Now that was something just repackaging something you've already heard. Now something completely new, TOPANAT. So this is a tool which leverages our integration of gene expression data and is unique to us because we're the only ones who integrate gene expression data like this. And it's similar to the gene ontology enrichment that most of you already know probably. So gene ontology enrichment is you have a list of genes and you submit it to either a web server or use an R package, and you find that your list of genes relative to what you expected from the genome are more often transcription factors or whatever. So more frequent annotation to some gene ontology terms. So here we do the same with anatomy. So each gene has expression in specific anatomical structure, these present absent codes that Frédéric just presented. And this is only possible because we have these present absent codes. So if I have a gene list, some anatomical structures are represented many times in the list. I have a gene list and many of these genes are expressed in the brain, for example. So I will have the term brain associated to these genes many times. For example, here I put the example frontal pole. I have 46 genes from a list which are expressed in the frontal pole. And here I have my gene list has 46 genes expressed in frontal pole, and my total size of my gene list is 46 plus 56, 112 genes. Is this more than I expect or less than I expect or what I expect? Would by chance 112 genes have 46 genes expressed in frontal pole? Well, I look at all the other genes in the genome and I see that out of 36,000, I have only 3,464 which are expressed in the frontal pole. And on this, you can do a test like a chi-square, a Fisher exact test. Here it's a Fisher exact test to look whether this is the random distribution you expect, and it is not. You would expect here 5.99 genes of a gene list of this size expressed in the frontal pole, and we get 46. That's 7.68 times more. And the p-value for a Fisher exact test for this is 10 to the minus 29, which is significant. I think we can all agree. So our gene list has much more genes expressed in the frontal pole, which is the part of the brain, than would be expected by chance. And you can do this generally for every anatomical structure. You would have your gene list, and for each anatomical structure, some genes will be expressed in this structure and some will not. And you have the other genes which will be expressed in the structure or not, and you'll be able to do a Fisher test or a hypogeometric test and find whether you have enrichment or depletion of your gene list in each anatomical structure. This is really like what you do for a gene ontology test, where you go over all the gene ontology terms and you look, do I have an enrichment of annotation of my gene list to this gene ontology term or not relative to the other genes, the background. So we have basically modified the TopGo package, which is a package for gene ontology enrichment, so that instead of using the gene ontology as an ontology, it uses UBRON as an ontology, and instead of using annotation of genes to gene ontology terms, it uses expression of genes in anatomical terms. And then we can do exactly the same statistics, including the fact that TopGo has deconvolution of the ontology graph, which basically means that the tests between different terms, since they are related, they are in a graph, is not independent. So genes, if you have your gene list, which is a lot expressed in cerebellum, it's also a lot expressed in brain. So these are not independent tests. So there's a deconvolution which is already it's a problem which was already solved in the package TopGo, because for the gene ontology it's the same. If I have a lot of hydrolases, obviously I have a lot of enzymes, and we can reuse this. It's the same mathematics and the same algorithms, and so all the deconvolutions which are possible in TopGo are possible in our modified version TopANAT. And so then we have both the web interface and R package, and here I ran the web interface a few days ago. I took all the genes which are annotated in Flybase. So Flybase is the model organ database for Drosophila, and they have annotations of knockout or knockdown mutants. So I took all the genes which, when you knock out or knock down the gene, you get reduced fertility. So this is information which is not from expression, it's from other information on the function of the genes. These are genes which, if you mutate them, you reduce the fertility of the fly. And I put them in TopANAT, and these are the terms I get. So you see that I get a lot of terms related to anatomical structures which are involved in reproduction: oocyte-associated follicle, nurse follicle cell, here a neuron, germline cell, Escherichia cell of the ovary, egg chamber, ovarian cell, progenitor cell in testis, Escherichia cell of post-embryonic organism, entofollicle cell in ovary, egg cell. So a lot of terms related to reproduction, also a few related to behavior, which will also sometimes in flies lead to, well, I mean to nervous system, so behavior, reduce fertility in flies. And we see that because we integrate all the data, here I have some single cell data. So this is a cell type in an organ. So this comes from single cell data. We have the fly cell atlas, I remind you, but also some of this comes from bulk. For example, egg chamber is something which comes from bulk RNA-Seq or microarrays. And it's all integrated together. So you get all this together. Here they are ranked by FDR because we do many tests. So we correct for the multiple testing, but then you can decide to take the not corrected p-value or the fold enrichment. Here, most of these terms are two-fold enriched. So we have twice as many genes in these structures as expected by chance. And these are reproducible and shareable. So if you would take this code and put it, you would get exactly the same gene list and the same results, and then you could modify the parameters to play with this and do it differently if you prefer to restrict to single cell data or restrict to adult or restrict to embryo and so on. Now, just something I should say in general about enrichment analysis, which is true also to gene ontology analysis. All these enrichment analyses, the background is critical. If you go back to this table here, the statistical test is comparing this to that. If you put the wrong thing here, you'll get the wrong conclusion on this test. So whether you do a gene ontology enrichment test or an anatomy expression enrichment test, it is very important that your other genes, your universe of possible genes, is the correct one. For example, if you have a gene list which by definition can only have protein coding genes, you should define your background as only protein coding genes and not include the non-coding genes, which are in Bgee, so will be included by default. If you had a gene list which came from, for example, you took genes which had a certain phenotype mutant in mouse and looked at the orthologs in human, your gene list can only be genes which have orthologs between mouse and human. So in that case, your background should be only genes which have orthologs between mouse and human, and so on and so forth. So it's very important whenever you do an enrichment test to have the proper background. And as an example, when we were testing TopANAT at the beginning, we found that we could get a bias just from the fact of which organs had been sampled for given experiments. So the sensitivity is such that if you would take all the genes which were studied in certain experiments, you would have a bias because these are the genes which were studied in some experiments, and so you have different organs. Another issue is that you have multiple testing. So I'll show you that we give here a false discovery rate control. This is a debated issue within the community of people who do such testing because false discovery rate assumes independence. These are not really independent since it's a graph. So some people prefer to use directly the p-value, some the FDR. In any case, you should be aware that you have done multiple testing. We have tens of thousands of terms in Uberon. There are also tens of thousands of terms in the gene ontology. Whenever you do this kind of test, you should be careful not to overinterpret if you have very limited p-value thresholds. And as I told you, the terms are not independent. I mean, here you see that many of these terms are coming from the ovary. So if there's high expression in the ovary, all these terms are going to come, right? So for this, TopGo provides you algorithms. The two we use the most are ELIM, which will only keep the most specific terms and just remove the others, and weight, which is what I used here, which is a bit more complicated way of weighting so that it prefers the more specific terms, but it will also give you the more general term, but downweighing them. And so just these concerns are general for enrichment analysis, and you should keep them in mind whenever you do this. But apart from that, I think TopANAT is quite powerful. In our experience, it usually works. And something I would like also to attract your attention to concerning TopANAT is that it is only on experimental data. So if you do a gene ontology enrichment in a non-model species, say platypus, you will get gene ontology results, but actually none of these were determined in platypus. No one did molecular biology experiments in platypus. If you do a TopANAT enrichment test in platypus, we will only give you results from gene expression which was experimentally measured in platypus. Now, this might seem a bit ridiculous example because not many of you are studying platypus, but even between human and mouse, there are biological differences. Humans are not mice, mice are not humans. A gene ontology enrichment will give you the same results because the annotations with very few exceptions are transferred between them. We will only give you from gene expression which was measured in human or measured in mouse. So the biological differences will be taken into account in this enrichment. And TopANAT is available on our website. You go to the top, it's there on the top of the page, and in the R package BGDB that Frédéric presented you, which both has much more options than the R package, than the website, and allows you to integrate it into your own workflow if you're working in R with transcriptomic data or with any other. Oh, sorry, I need to drink. With any other type of large-scale genomic data, if you want at a given step to run TopANAT, you can directly include it into your R workflow. Another type of tools we provide are tools around homology. So most of you know about autology and homology of genes. So you have autologous genes between, say, human, mouse, fly, and so on. When we speak of gene expression, we speak of gene expression in an anatomical structure. So if I have autologous genes between, say, human and fly, and I want to compare the expression, I have to compare the expression in organs which are homologous, which I can compare. And this is non-trivial. There are bioinformatic methods to give you orthologs. There are no bioinformatic methods to give you homologs. So we annotate this ourselves. So the biocurators of Bgee annotate anatomical homology, and this is entirely manually curated from the literature, Evo-Devo literature, paleontological literature, and so on, comparative anatomy books. It is not derived from the gene expression. Otherwise, this would be circular. We don't take the gene expression from Bgee to define homology of anatomy. We define homology of anatomy separately, and then we can look at gene expression in the homologous structures. And we provide this information first directly at the anatomical level. So we have a tool where you can put anatomical identifiers. So here I put all the tissues or cell types which were sampled in the Bgee Tech experiment. And I ask for homology between human and zebrafish. Now, zebrafish is a vertebrate like humans, so there's quite a bit of anatomical homologous structures, but fish and humans are quite different. It's not another mammal, it's a fish. So there are differences also. So here what you see is that you have 31 of these structures which have homology, and all those which don't have homology, because there's not a homolog structure between the human and the fish. And those which do have homology, sometimes not the same term. For example, heart left ventricle or primary heart field, or here lung in human. There are no lungs in zebrafish. Zebrafish don't breathe air, but they have a swim bladder, which they fill with air to go up and down in the water. And this is the homolog actually of the lung. So you get this information manually curated automatically from this tool. And as we showed you towards the beginning, you can also use this homology to compare gene expression between species. So you have autologous genes or other genes you want to compare between species. You're going to be able to compare their gene expression based on the homology of anatomy. So for example, here you have genes from a brain-specific gene. So I took the one of the examples from the expression comparison page. You go on this expression comparison page, you put here gene identifiers, which you can link directly from the ortholog or paralog field in a gene page, or you can put your own gene list from your own analysis, and it will give you the anatomical structure which are homologous between the species represented here, with the level of expression, how many genes are found in this structure, how many genes have no data in this structure, because different structures have been studied in different species. So you might have more species, for example, where the cerebellum has been studied, so you have 9, but less species where the cerebral cortex has been studied. But those where it has been studied has very high expression, so it will give you both of these. This brain-specific gene has conserved expression in the cerebral cortex, in the cerebellum, in the metencephalon, and so on. So that's the conclusion of the lectures part. Then we will go to questions. So Bgee, as we told you, is I think we showed you how we want to understand gene expression to make it useful. You saw all those things where we try to understand the different anatomical structure, different cell types, the different protocols. What does it mean that the gene is expressed? How can we call a gene present? How can we normalize? How can we compare? We have to spend all this time understanding gene expression, understanding the biology which comes from, to make Bgee useful with the kind of tools I showed you at the end. I think that's it for this. So I will stop sharing my screen. I see there's several points here in the Zoom. For the expression comparison, how many genes are accepted when I use the web server?
Answer that, but there is another question. I did not get where are the additional tools you mentioned. Maybe I can share my screen quickly.
Yeah, go ahead.
Going on Bgee. So top anat is here, expression comparison is here. So top anat, here you get examples when you click on the link. So yeah, so you enter a list of genes here. You select various parameters, and at the end you get the list of enriched terms with the p-value, the FDR, the fold enrichment as Marc presented. And still at the top, you get expression comparison and you get examples here. So it takes a while. So this page, we are going to update it quite a lot in the next few months. So it's currently slow. So yeah, I have to end for that query to end. But yeah, it's based on gene IDs as we discussed in the chat. Yeah. So you really have to enter IDs. It doesn't work on gene names because managing gene names in multiple species is a bit complicated, because if you just put the name of a gene, we won't know which species it comes from. So we need to improve that interface. But yeah, that's what Marc showed you here. So this is a list of genes that is brain specific, and you get brain specific term at the top with various coverage between species, with subspecies where we don't have the data in the structure. So yeah, it's all at the top. And for the homology, yeah, we just highlight the most important tools in that menu here, but then you can find really all the tools here in this complete menu. So you get also the anatomical autology. So you get in tools, you get top anat, expression comparison, and anatomical homology that Marc presented, where you have to select the species for which you want to retrieve a homology, and with those examples like Okay, give me the homologue of lung in human, give me that in zebrafish. So again, you see the same interface as for expression present, it's based on the same code. So this is a bit too optimized. So you can see here, like, yeah, the homologous tissue of lung in human, in zebrafish, this is a swim bladder.
So thank you all for attending and for participating, with the woo claps and the votes and everything. That was very nice. So we look forward to continuing to develop Bgee and to continue to work with our users. We hope you learned something new or enjoyed yourselves.